everyone, it's Mrs. Shannon, and sorry the screen is black, but I was just changing this into presentation mode. I'm going to kind of review DNA structure and function a little bit and talk about replication. This presentation may be helpful when you're doing the POGL if you had some questions, especially when you're looking at Model 2. And I'm also going to talk a little bit about Model 3 and the expectations for that. Okay, so just as a reminder, DNA's function is basically to store, copy, and transmit genetic info. It is the master blueprint, and this genetic information encodes for making proteins. Those proteins are going to go out and do all the work. Now, it is the DNA that this strand right here that you'll see, the double helix, the two-sided ladder, the thing that you made, that is a nucleic acid. That's what the N and the A stand for in DNA. It's a polymer because it's made of many smaller subunits, monomers, and they're called nucleotides. Now, just as a reminder, there are 4 biomolecules that we talked about. We've talked about carbohydrates for a little bit. It's the primary source of quick usable energy. We talked about it in photosynthesis and cellular respiration. Lipids store energy long term. It takes more energy to break those bonds, but they can release a lot of energy in the process. The body doesn't like to do it because it takes so much input of energy initially. So it saves it and tries to use up carbohydrates first. And it also offers protection. Remember, we talked about the cell membrane having those phospholipids. And so it offered protection for the cell, kind of as the gatekeeper, allowing what comes in and what goes out. And it also can add insulation if you're looking at it on a macro level when you're looking at organisms. Nucleic acids are what we're talking about right now. It's the genetic code. It creates the proteins to do the work with this code. The one thing that we haven't really talked about, and this is like my favorite biomolecule are the proteins, and these are the minions of the body. And so when we say that they are the minions of the body, they do everything that their boss, which is the DNA, tells them to do, okay? So they receive the instructions from Groot or the DNA, and then they go out to the body, and maybe their job is to break down food. Maybe their job is to help build new phospholipids. Maybe their job is to help other molecules enter the cell membrane. Whatever it is, they are just the minions. They receive the instructions from the DNA. We talked about protein channels just recently. We're also going to talk about another one called enzymes coming up. And we're going to talk about how the DNA encodes to make these proteins that go out and do things. And so we'll look at various other specific proteins in the process to see like kind of the vast spectrum that there are in the body. So just as a reminder, basic structure of DNA, and you explored this when you made the model. And then when you were looking, when I say the model, the pop bean model, and then when you were looking at the POGL, model 1 on the POGL covered some of this too. DNA stands for deoxyribonucleic acid. It's not the only nucleic acid. There's also something called RNA, ribonucleic acid. The big difference is the sugar and the type of bases used, as well as it being single-stranded for RNA. But we'll come to RNA when we start looking at how this information in the DNA codes for the making of proteins. Now, DNA is double-stranded, and when we say that, it means it has two sides of the ladder. Now, you guys, when you made the ladder and you made the connections, hopefully you saw that the pop beads were actually kind of flexible, and we could have kind of twisted it into this double-stranded double helix shape. Remember, the discovery of the shape was made by James Watson, Francis Crick, and also Millikan with the help of Rosalind Franklin's crystallography picture. Okay, now this part right here, you guys probably looked at the book to form these nucleotides when you were making your own pop bean model, but it's also listed in part 1. Those monomers, those nucleotides have 3 parts. They have a nitrogen base, okay? That just means that they have a nitrogen in them. They have 5-carbon sugar, and they have, oh, right there's the sugar, and they have a phosphate group. And so here we're going to, later on, there's just a small difference between ribose and deoxyribose, and you can see it's the presence right here of this little hydroxyl group. Okay, so here is 1, 2, 3. There's one base. 1, 2, 3. Another base. 1, 2, 3. A third base. 1, 2, 3. A fourth base. Notice that some of these bases have 2 rings attached. Some of them have just 1. And the reason is because they're divided into 2 categories. You do not need to memorize purines and pyrimidines, okay? You don't need to memorize the structure of them, just to recognize that there are 2 categories. So I'm going to skip through this slide a little bit quickly. You will come to this more so a little bit in AP Bio, but the whole thing here is A combined with T because of the size here. Up 2-ring one will always bind with a 1-ring one. Okay, guanine to cytosine, and we'll talk about uracil and RNA later on. Okay, so this probably looks pretty similar to the model that was in the POGL. What I want you guys to notice is that there are hydrogen bonds. Now, when you made between the bases of each side of the ladder, when you made your pop bead one, you only used 1 hydrogen bond to represent it because it was a model, and the idea was just an overview of the structure, not to be exact. You don't have to know how many are between each base, but I will tell you that what you do need to know is that these hydrogen bonds, they're calling them a bond, but it's really not a bond like an ionic bond or a covalent bond. It's not as strong. It's a pretty weak bond, okay? And it's just kind of this, more like an attractive force, if anything, between the bases. Whoops, and I thought I had it here. Oh, right. And don't forget complementary base pair, Chargaff's rule. A always goes with T, G always goes with C. And I remember it as apples are found in the tree, cars are found in the garage. So if that helps you remember it, feel free to use it. I think that's what Amoeba Sisters uses too. Again, don't remember how many they have between them, but do know that those connections, those bonds, those loose bonds between them are called hydrogen bonds. And that's important for when we look at replication and something down the line called protein synthesis. So that's the bases are what connect it. And here you can see the bonds. But on the side, they call this the backbone of the ladder. Okay, and it's just the sides of the ladder. So if you hear backbone, it just means the sides of the ladder. They usually refer to the bases as the steps of the ladder. And so when you're looking here, you can kind of see that. Here's the bases connected, the steps, and here is the backbone. So here was a picture of the first page of your POGL. So again, I just wanted to show you the 3 parts of the nucleotide. The nucleotides make up the sides. You can see that it's double-sided. You can see the same pattern and what matches up. If we were to twist this, we could actually turn it into a helix. It just means twisted ladder of DNA. Okay. Now, the next part is DNA replication, and this means copying. And when we replicate, we make exact copies, okay? Exact copies. Again, you want to make exact copies because it's part of the cell cycle when cells divide during mitosis to make exact replicas. So DNA replication is copying and synthesizing new DNA. Again, it occurs in the nucleus before a cell divides. This kind of goes a little bit into Model 3. Model 3 is also going to talk about chromosome structure, which I'm going to wait and talk about in a different screencast simply because I want some props to show you that. But it occurs in the synthesis phase, and it occurs before a cell divides so that each new cell has a complete set of chromosomes. We have 46 chromosomes in our body cells. Our body cells are known as somatic cells. This is different than our gametes. Our gametes are sperm and egg. We'll find out in a process called meiosis that it's slightly different, and that meiosis produces cells that are different than their parents and have half the number of chromosomes. So each of our sperm and egg will, depending on if you're a boy or a girl, have 23 chromosomes. But we'll get into that later on. For this, we're going to focus on body cells, somatic cells. So we have 46 chromosomes, 23 from mom, 23 from dad. And replication is going to take place here. When that copying is made of exact replicas, you will then have 92, technically, chromosomes. But that's good because then when it goes through mitosis and divides, each cell will end up with 46, just like it started with. So how does DNA replication begin? Well, first of all, we have to separate this DNA strand. It begins at the origin of replication, and it begins to fork at a fork. We call it a fork because it's like walking down a path and you come to a fork in the road. Which way are you going to go? So they open, forming Y-shaped regions, and that allows new strands to grow. Now there's space for new strands to grow. So we have to unconnect it from the parent molecule, or unconnect the parent molecule, so we can start forming replicated DNA at each side. So looking at this model here, hopefully you see some similarities in the replication fork, and here's our replication fork here. You had to identify kind of the steps of replication. And so we're going to go through that. And we're also going to talk about this helicase molecule too a little bit. So really quickly, there are certain proteins which were made by DNA previously, that are involved in working, because that's what proteins or minions do. They work to help DNA replicate. One of them is helicase. It unwinds the double helix, and it unwinds it by breaking apart those hydrogen bonds. It just goes down the DNA strand and it unzips the DNA strands by weakening and loosening those hydrogen bonds so the nucleotide bases break apart. Okay, so helicase unwinds the double helix. It unzips it. And you can remember helicase begins with H, just like hydrogen begins with H in hydrogen bonds. So it unzips the hydrogen bonds. Another big player here is DNA polymerase. DNA polymerase is going to bind nucleotides. It's going to help when the nucleotides come in, it's going to help bond them together here, polymerize it, make a new polymer. And then there's ligase. Ligase, it joins something called Okazaki fragments, and I might have even deleted that from this slide. Basically, this happens in parts, and you're going to have like all these little parts of replicated DNA that need to be joined together, and it's like the glue. Ligase is going to like kind of seal them together. The ones that I'm concerned about here are these two, helicase and DNA polymerase. Okay? Notice, and I'll come back to this when we talk more about enzymes and proteins when we do protein synthesis, that all of these end in ase. This is a sign that these are enzymes. Remember how we talked about ose and like glucose, fructose, sucrose being a way that we can identify molecules as being carbohydrates? Well, now we can identify molecules as being enzymes because they end in ase. So that is a note that you will definitely want to make somewhere, because if you don't know what something is, but you see that it ends in ase, you know it's probably an enzyme and it probably does something. We will spend more time again on enzymes when we talk about proteins and protein synthesis. But for those that are really wanting to know what this word is that I'm talking about, enzymes are proteins that help reactions occur, help speed up things so they happen in the body. So the first step is that replication fork. Helicase unzips the DNA down the middle by breaking the hydrogen bonds. That has to happen first. Then before DNA strands can form, there must be some RNA primers. You don't need to remember RNA primers. It's just to get it ready. But what you do need to see is that these nucleotides, there's free bases, okay? There's A's and G's and C's and T's, and they're just kind of randomly floating around in the nucleus, okay? But they're going to be able to fit in now and find their complement. If this is an A, it found a T here to connect with, okay? So these free nucleotides, once it's unzipped, are able to come in. So going back, helicase unzips the DNA, and then that DNA can split apart. Now there's room for the nucleotides to come in. Again, ignore the purple RNA primer part. Okay, now that we have these nucleotides, though, the polymerase moves along and base pairs nucleotides to each original parent strand. So it helps those new nucleotides, like these green ones, connect with the bases of the original parent strand by reconnecting hydrogen bonds. Okay, so helicase unzipped them. Polymerase is going to go through and reform them. So here, you should be able to go through here and figure out we have to have our hydrogen bonds. Before we can split apart, we need our hydrogen bonds broken. We have a special molecule here, an enzyme. Then it's able to split apart. We have all these free nucleotides just floating around. They're able to now have room to come in. And then the polymerase is going to come in and help reform these bonds. And then you'll have your new strands. So hopefully the previous slides and me just kind of working through this, I don't want to give answers completely away, but helped with number 11 as well as number 12. Now, the idea here is I want to throw this out there too, because you may come across it. When they proposed this model and how it made copies of itself, Watson and Crick did this idea of semi-conservative model for replication, in which case they conserved each new strand had one of the original parent strands. So you have your parental DNA. Your new DNA here has the top copy in this picture of the original DNA, and the new DNA formed by nucleotides that came in. And then this second strand here of DNA has the bottom parental strand, and this pink one here formed by matching complementary nucleotides that were floating around coming in. So you started with one, you end up with two, and each of the new ones has one original parent strand or side. I'll call it a side in it. Now, the whole idea of mutations here and why I'm having you guys watch the mutations one is by Amoeba Sisters, is that Amoeba Sisters one covers mutations in more detail because it covers gene mutations as well as chromosomal mutations. We're only talking about gene mutations right now. We're going to come back with chromosomal mutations when we talk about that process I talked about earlier, meiosis. But when we're talking about gene mutations, we're talking about DNA mutations. Remember, DNA is just a strand of instructions, and there's different genes. Here's one gene right here. Well, actually, let's consider it like one gene would be from here to here. And that means it's one set of instructions for one specific protein that goes out and does one specific job. It occurs when a base, such as A, T, C, or G, is accidentally changed. Now, when it's accidentally changed, remember that polymerase that came in and helped bind the nucleotides together, the free nucleotides that were in the nucleus matched up, and then the polymerase came in to help them bind again? Well, it also proofreads to catch most of these mutations, and it can fix it. It can keep it from bonding to the DNA. That's really important with checkpoints. The checkpoint leaving out of synthesis. And it's a way for organisms to help maintain homeostasis. But it only catches most of these mutations, not all of them. Some of these mutations can be beneficial, some of these mutations can be harmful, and some of these mutations can have no effect at all. Some causes besides DNA polymerase, some mutations besides just accidentally being changed here can be from the environment, UV rays, chemicals, or X-rays. As a result, you can have genetic disorders or cancer even. We'll talk about genetic disorders because that's going to be more when it's in your gametes, when it involves your gametes, because that means it can be passed on. If it can't be passed on, if it's only in your somatic cells, like skin cancer, for instance, then that's not considered a genetic disorder, although you may have a gene that predisposes you, and that gets into something else completely. So I think that's the end of the slideshow. Keep up with the Amoeba Sisters. I'm going to keep putting notes in as well. These notes are going to be optional. And just to let you know, once a week, at least once a week, if you want me to do more, I will be holding a Google Meet during office hours to answer questions or just talk about whatever you want to talk about. Right now, I'm kind of holding off on setting up a definite schedule because, as you know, like things keep changing. Now we're going to be closed longer than we thought we were going to be closed. And so we're really trying to, they just put out a calendar yesterday of which days are going to be e-learning and which days are going to be waived. And so I'm really trying to get something more secure in place before I start pushing out times and hours and expectations and even future assignments until I get a better idea from Concord about what e-learning is going to be looking like in the next couple of weeks. But feel free, message me anytime with anything because I miss you guys. And I will talk to you later.